Hi guys, I think we'll make a start. Sorry, I'm just starting slightly late and just having some problems with my slides. I hope everyone's doing okay. I know your exams are coming up soon. You're probably quite stressed, but hopefully this lecture will just provide you a bit of an overview on the topic of virology. Going to be mainly covering kind of like part A, high yield stuff, and then also going into a little bit of depth on like quite a common part B exam question on comparing HIV and influenza. So hopefully it'll be useful. Um, it's my first time giving like a lecture like this, so it'd be great if you could give some feedback at the end as well. There's a QR code to scan. That would be great. Let me just share my slides with you. Yeah, I hope you can see the slides at the moment. Please just say something in the chat if you can't. And also, at any point, if you have any questions throughout, feel free to just unmute yourself or write the question in the chat or email me afterwards. I'd be really happy to answer any questions as well. Okay, yeah. So here we go. So be about an hour today, hopefully a little bit less. I just want to go through the structure of viruses, how they're classified, some key definitions that you might need for the exams. Like I said, comparing HIV with influenza. And then at the end, I put some part A multiple choice questions for you to have a look at. We might not get time to go through them and the answers today, but it should be useful nonetheless. Okay, so this is just some sections from the syllabus. Um, there's quite a lot actually in the syllabus on viruses, but I've just highlighted some of the key points, which I think are probably most important for the exams, and hopefully we'll cover most of that today. We're not really going to cover vaccines and viral treatment as much. I'll try and do a little bit here and there, but it's probably not going to be within this lecture. I'm mainly going to focus on kind of like structure, classification, and how they enter into host cells. Okay. So I'm sure that you've seen something like this before, but this is just kind of the general structure of a virus. The one I'm showing here is influenza. And generally they have a capsid, just the outer layer, a protein coat, lipid membrane, some kind of genetic material within them. And then viruses can be enveloped or non-enveloped. Generally, the non-enveloped ones tend to be more resistant to things like heat, acid, like adverse environmental conditions. And the non-enveloped things are often spread through like the fecal-oral route. Enveloped viruses tend to be respiratory viruses. Um, and the envelope is just kind of like a layer of protein around the outside, which is glycosylated. And then also really importantly for viruses, we think about attachment and entry protein. So how do they actually get into the host cells? So heparan sulfate is an example of a protein which is on the surface of cells, and then as a sialic acid. And for example, in influenza, it's the hemagglutinin which binds onto the sialic acid, which allows it to enter into the cell. So that's really important when we think about like therapeutic targets as well. Okay, so this is quite like a big table. I'm sure you've seen this before as well in the lectures. But this is just it's something called the Baltimore classification system. So from 1 to 7, and it just classifies viruses based on the type of genetic material, how they replicate. And then I've given some examples as well. The ones highlighted in red are kind of the most medically relevant ones that you might want to focus on. Um, but just to go through, like group one is double-stranded DNA, can be positive or negative sense. Um, replicates in the nucleus, requires host polymerases. And then sometimes those viruses can go on to cause cancer as they force the cell into the cell cycle. And that's adenoviruses, herpes viruses, and pox viruses. Group two is single-stranded DNA in the positive sense, which converts double-stranded before RNA, and that replicates mainly through nuclear replication. Parvoviruses there, so nothing really that medically relevant. Group three is double, sorry, group three is double-stranded RNA can be positive or negative sense. Group four, positive sense, single-stranded RNA, which generates a negative single-stranded template. I'm sure that you can read the rest of that. It's probably not the most helpful, me just reading it out to you. But I put this in, so maybe when you go back through these slides, if you want to, before the exam, you can kind of have a look at this one and like test yourself and see how much you can remember out onto this empty grid. So just going through some key definitions as well, might be quite useful for the multiple choice exams. The multiplicity of infection is the number of infectious particles per cell. Tropism is the permissive cell or tissue, so which host cell the virus actually infects. So, for example, in influenza, we're thinking about like upper respiratory tract, um, that kind of thing. With HIV, it localizes to gut-associated lymphoid tissue, among other places. The eclipse phase is the period after an infection, but before any new virus is produced. The burst size is the number of new infectious particles produced by an infected cell. So obviously, the larger that number, the more damage it's going to do. And then a defective virus is a non-infectious particle. And again, I've just left those blanks, so if you go through it, you can test yourself with that. So I'm sure you know as well, there's a variety of different ways in which viruses can have an effect within the body. So whether that be an acute infection, which is short term and easily resolved, or something like so that would be something like influenza for most healthy people is a short term, maybe a week or two, and then that's cleared from the body with an adequate immune response. In something like herpes, which looking at the third one down, herpes simplex, so HSV1 causes cold sores, which is latent within the ganglion around the lips, and normally is repressed in most people, but then during times of stress, for example, malnutrition, emotional, physical stress, that can recur due to immunodeficiency. Um, and then looking at the bottom one would be something like HIV, where there's an acute phase, and then it goes into latency for many years, can be up to 10 years. And then it obviously during that time causes a lot of damage to the immune system, and then eventually causes AIDS. Another key kind of like family of viruses which you should know about is hepatitis. Um, so these all infect the liver, and the symptoms of acute hepatitis are things like nausea, vomiting, hepatomegaly, so an enlarged liver, pale stool, dark urine, and raised ALT, which is alanine transaminase, I think, which is just like a biomarker of liver damage. So you don't need to know too much about what it does in metabolism, but it's raised when there's damage to the liver, if it's inflamed or if there's an infection. And then again, I've just written out kind of like the key points from each virus to be aware of. You probably all had like a Hep B injection, I think, before you started. So yeah, there's Hep A to E. Hep A is spread through the fecal-oral route mainly. It's not actually normally vaccinated against in this country because there's quite a low instance of it, but it's more common in certain populations. Generally through like fecal-oral routes or contaminated food, contaminated water, that kind of thing. Generally, in most people, it's quite mild symptoms, and it's non-chronic. Hep B can be spread through kind of like body fluids, sharing needles, mother to child. Um, the same with Hep C. Hep D generally only occurs in combination with Hep B, and then Hep E as well is also mainly found in pigs, and is spread through the fecal oral route. Generally mild, but can be very dangerous in pregnancy. So I think it's important as well that you know the symptoms of acute hepatitis. So those are quite often asked in exams that are at the top of the screen, and then also some of the routes of infection of the others. And then as well, this again is just like classic Part A questions. The different types of viruses that can cause tumors. So um, I think the first one ever identified was Roost sarcoma virus in chickens, if I remember rightly. And then since then, we've discovered a huge number of different viruses which lead to tumors. The key ones are highlighted here. So HHV4 is human herpes virus, also called Epstein-Barr virus, which can cause most commonly nasopharyngeal carcinoma and also lymphoma. HHV8 um, is causes Kaposi's sarcoma, and that is the causative agent, but that cancer is almost exclusively seen in people with HIV. So you kind of have to have HIV plus a HHV8 infection to then end up with Kaposi sarcoma. HBV and HCV, so hepatitis B and C, can cause hepatocellular carcinoma in some people. HPV, there are so many different strains of HPV. The most dangerous ones are probably 16 and 18, have been really linked to cervical cancer, but other cancers, including anogenital papilloma, head and neck carcinoma as well. We have actually got quite a good virus against HPV now, guys, so I think which can protect against a large number of strains. So that's really good. HTLV1 can cause adult T cell leukemia and Merkel cell polyomavirus causes Merkel cell carcinoma. So those are just really important things to know, I think, as well. And again, I've left the empty grid for you to kind of like test your knowledge on that afterwards if you look back through this. So now I'm just going to focus on two quite common viruses, well, quite commonly asked about viruses, in a bit more detail. So going to be looking at influenza and HIV and comparing those two, their patterns of infection, etc. So influenza can be like seasonal, or it can also cause pandemics. Um, it's kind of depending on the populations they're in. The different subtypes are defined by their hemagglutinin and neuraminidase molecules. So it'll have like I think there's 11 different 18 types of HA and 11 of NA, well, I think. Um, and they can cause infection in a number of ways. And when we think about kind of like big pandemics, it's normally due to antigenic shift as opposed to drift. So the difference between those is that antigenic drift is kind of small genetic changes within the makeup of the virus, whereas antigenic shift is normally when two different strains mix the genetic material and create something which is completely new, and therefore the body does not have immunity to that anymore. Um, that's why kind of the vaccines for influenza are developed yearly, based on the emerging trends and patterns within infection. It's generally spread through the upper respiratory tract through respiratory droplets, and we, like I said, we do have a really effective vaccine for that. Um, and then just showing you the like general structure of it again. The key things is neuraminidase and hemagglutinin because hemagglutinin is important for entry into the host cell. Neuraminidase is important for release of the virus from the infected cell. And then there's some important channels there, M1 and M2. Um, but we'll cover this in a bit more detail over the next slides. And influenza is enveloped as well. It's got a protein envelope. So this is just kind of like a general overview of the entry, replication, and release of influenza into a cell. You can see it binds to sialic acid receptors on the host cell. It's endocytosed. Really importantly, with influenza, there's acidification and membrane fusion. So that's pH dependent, which is not the case in HIV, which we'll talk about in a second. You then get nuclear import, transcription, replication, translation, and eventually the virus is released. Using neuraminidase to cleave it. And then this is the kind of equivalent in HIV. So HIV fuses to the host cell surface. GP120 needs to bind to CD4 receptors in conjunction with CCR5 or CXCR4. The HIV RNA is then enters the host cell, undergoes replication, reverse transcription, imported. Viral DNA is transported across the nucleus, integrates into the host DNA. This new RNA is then used to make viral proteins, which can then leave the cell. So I'm just now going to like break down each step in that in a bit more detail, comparing them to each other. This might be asked like as an essay, you might want to answer this. It's probably a bit more depth than would be for multiple choice. Um, so influenza needs to attach to the target host cell, which determines the tropism, so the permissive cell or tissue, normally upper respiratory tract. And as I said a few times, it binds to sialic acid with hemagglutinin. That's like the key thing to know. Um, and then kind of there's different types of sialic acid which are expressed, and then there's differences in species as well. So humans, there's alpha 2,6 linkage on the hemagglutinin receptor, which is the key target, whereas in avian influenza, it targets the alpha 2,3 linkage. Um, Often as well, there can be like species which act as mixing vessels that allow exchange of genetic material between different influenza viruses and the kind of development of new strains. And sometimes that's in pigs, or often like a mixing vessel. Um, HIV also attaches to cell surface receptors, but so it is specific. So it's CD4 receptors and then CCR5 and CXCR4 on T cells. And it needs that the CCR5 or the CXCR4 as a co-receptor. There's also sometimes some non-specific binding of HIV, but that's less important. And then the next step is endocytosis. So the virus is engulfed in an endosome in influenza, which relies on acidification um, to allow for fusion and entry. The envelope fuses and occurs at the cell surface, forms an endosome which is acidified. In HIV, there is also endosome fusion, but it's not dependent on low pH, and the envelope is degraded by proteases. So in influenza, the acidification of membrane fusion leads to protons entering the endosome, conformational change in the hemagglutinin, which exposes a fusion peptide. The hemagglutinin domain moves in low pH, and the alpha helix extends, which exposes a hydrophobic peptide. This peptide inserts into the endosome membrane and brings the two membranes together to fuse and release into the cytoplasm. This requires the hemagglutinin to be first cleaved. Um, and then there's monobasic, so cleaved by a limited number of proteases just at the lungs. So that would be like a quite a localized infection, or multibasic, where you get systemic infection in a number of different tissues, which is more serious. The acidification occurs via M2 ion channels, and then that's quite a key target for drugs. So M2 ion channel inhibition through a drug called rimonatidine prevents the release into the cytoplasm. HIV, in terms of entry into the cell, there's uncoating of the core to create a pre-integration complex, which then gets into the cytoplasm. So it's a lot less complex than influenza, and it's not dependent on pH. Then the genetic material. In influenza, there's nuclear imports of the viral ribonuclear protein through the nuclear pore, which is active transport, and that uses the cell's own importin to transport it into the nucleus. In HIV, there's no nuclear import, so the reverse transcription occurs outside the nucleus in the cytoplasm. There's a few kind of reverse transcriptase steps to prime the binding site, which initiates transcription from 3' to 5', forming a single-stranded DNA, negative strand. RNase then cleaves RNA. The new DNA strand circularizes, which hybridizes with a repeat sequence at the 3' end of single-stranded RNA. The reverse transcriptase hydrolyzes remaining RNA, leaving only the polypurine trapped in place. This is the primer to initiate synthesis of the second strand of DNA, which then the reverse transcriptase elongates this to form double-stranded DNA, and then RNase removes everything. In terms of, sorry, in terms of transcription, influenza is a group 5 Baltimore classification, so it's negative strand single-stranded RNA, but influenza is an exception to kind of what happens mostly in that group, because replication occurs usually in the cytoplasm, but occurs in the nucleus in this case. So it must be copied into the opposite sense, so positive sense strand by RNA polymerase first, which is unique to influenza in this group 5 class. This step is also another target for antivirals, so favipiravir is a drug which is in development to inhibit the active site of this. This ends up forming mRNA, um, and the virus has an mRNA polymerase to synthesize a cap structure which the host recognizes as its own, which allows effective integration. In HIV, the proviral genomic DNA is injected into the nucleus, integrated by an integrase into the host cell genome. Um, then a series of steps initiate transcription, which also transcribes the viral genome, which is embedded. And then in HIV, there's a gene called TAT, which enhances transcription. So just in terms of translation, in influenza, there's 8 genome segments encode 10 major polypeptides. In HIV, the viral proteins are transcribed as precursors generally, so they require protease cleavage to form the mature protein. And then in terms of assembly of the virus, influenza, there's a concerted assembly, so a capsid forms around the genome, and then you get nuclear export of the viral ribonuclear protein. The M1, which is the matrix protein, has nuclear export protein bound, and that is recognized by a host molecule to exit the cytoplasm. In HIV, there's assembly of a viral protein and the genome, and there's a concerted assembly, so a capsid forms around the genome, and then another HIV protein called VIF stabilizes the newly synthesized viral DNA and transports the virion core through the cytoplasm out to the membrane. then kind of the release of the virus is a key step, which allows the virus to go on and infect new cells. If this didn't happen, then the virus really wouldn't be very infectious or dangerous, but this is a really key step and also a big target for drugs as well. So in influenza, the virus is released by budding, which happens in a lot of different viruses. It takes a piece of cellular membrane with it and forms um, like a vesicle, which buds off and then goes on to affect another cell. And the key step in this is neuraminidase, which cleaves the link between the sialic acid and the receptor, so it allows the virus to be released. That's really important to remember. And a drug target here would be neuraminidase inhibitors, such as Tamiflu. Sorry. In terms of HIV, for the release, there's viral budding again, but it's released as free particles. Um, and a protein called VPU enhances the release of HIV virions from the cell. It does this by antagonizing tethering, which is an interferon regulated host restriction factor that cross-links virions on the host cell surface. And then just going to talk a little bit about how each of these cause disease, basically. Um, so influenza is reliant on host factors at multiple stages of replication for entry and also for how it replicates within the cell. The virus replicating causes damage to cells because as it is released, the cells are damaged, and also cells die in the process. And mainly that affects the epithelium and the bronchus and bronchioli. So cells of the upper respiratory tract where it actually infects in the first place. Um, the damage of ciliated epithelium in the airways does also allow bacteria more easily to pass into the respiratory tract. So often you get like a co-infection with bacteria as well. As obviously like the mucociliary escalator is not functioning as well. Virus replication causes induction of interferons, cytokines, and other kind of soluble response factors from the immune system, the host immune system, which cause that kind of common flu syndrome, so fever, headache, chills, malaise, um, so things like TNF, etc. Having a segmented genome is really advantageous for influenza. Um, through evolution, it's been able to gain different segments from different viruses, and that's how pandemics and antigenic shift occur. The virus needs to have a copy of each segment as well. So it has what's called like a packing problem, but it overcomes that by collecting complexes, and then there's like a likelihood that a certain percentage will have one in each segment. This, if you want to add some experimental evidence as well, there's some electron microscopy experiments um, shows that the virus has a way to package each of these different components together. Um, in terms of HIV, it has non-specific immune activation in the first instance. It leads to CD4 T helper cell death. Um, so the actual infection drives apoptosis in the cells. Uh, Also, like a lack of signaling from the T helper cells means that there's also a drop in B cell number. There's a lack of mucosal barrier function with Th17 cells and also immune regulation from Treg cells. You get exhaustion of CD8 cells, and then HIV forms these latent reservoir in memory T cells as well. Um, as I said, HIV-1 homes to the gout, which is gut-associated lymphoid tissue, tends to be a big reservoir of that. Um, and HIV can persist because basically it inactivates the host immune system and also has such extensive mechanisms which allow big antigenic variation. So, for example, its reverse transcriptase has no proofreading mechanism. You also can get a recombination between different HIV genomes. You can get things called chimeric viruses, which are kind of a fusion of two different types of HIV as well. Um, there's deaminase hypermutation, which is an enzyme which actively induces mutations within the HIV um, genome. And that's why kind of antiretroviral therapy has to target a lot of different angles, which also means, well, this huge kind of antigenic variation means that a vaccine has so far been really unsuccessful. Um, yeah, hence combination therapy. This is the So influenza, we do have some quite good vaccines for. It's an inactivated vaccine, the main component being hemagglutinin and neuraminidase. It can be kind of a purified subunit vaccine or a whole virus, which has been attenuated, and it induces antibody humoral immunity. You also get a live attenuated virus. Um, Body temperature is too high for good replication, but some does occur, so it induces humoral and cellular immunity. As it says there as well, you develop a new vaccine each year from the World Health Organization in response to the antigenic profile of the virus in that year. Normally, they're trivalent vaccines. They have three components. Um, yeah, and as I said on the slide as well, it's hard to have kind of an influenza vaccine which would carry on from year to year based on the antigenic variation as well. Although there are some regions of hemagglutinin which are quite conserved between different subtypes. So that would be perhaps an avenue to make something which can be um, effective more long term. And as well, there's antiviral drugs, which I've mentioned before, which target different steps of the entry and release process. HIV. So there have been some kind of experimental attempts to develop vaccines, including like inactivated subunit and vectored vaccines. Most of these have been unsuccessful so far. I think there's a few different strategies. For example, like kick and kill was one. Um, so kind of getting the HIV out of where it is stored long term in latency and trying to mount an effective immune response against it. Um, we do, however, have quite good therapy for HIV for treatment and also for prevention. So it's something called combination antiretroviral therapy, which uses reverse transcriptase inhibitors in combination, so like nucleoside analogs, um, azithromycin or nevirapine, which are direct reverse transcriptase inhibitors, along with protease inhibitors, so sequanavir, which blocks the maturation of the HIV proteins, and also integrase inhibitors, which block DNA integration. Um, you can get antagonists of CCR5 as well, which inhibit HIV entry. Um, Marovaroc is a selective antagonist of CCR5. Um, so they were all kind of like therapies, although once somebody has the HIV infection, at the moment it can't be cured. So the main focus is on preventing that through the use of PrEP, which is called pre-exposure, which is stands for pre-exposure prophylaxis, which is given to patients who are at particularly high risk of developing HIV. And it's a combination of the above drugs, essentially. Um, there's some experiments, so Cohen et al. 2011 reported that the use of antiretrovirals decreased the viral load sufficiently to reduce transmission of HIV by about 96%. So patients can achieve almost undetectable levels in the blood, which means that the chance of them passing that on to somebody else is much lower. Okay, I've got through this a bit quicker than I thought I would. Um, but I've just got some like part A style MCQ questions, which I don't know if you want to have a look at. I think we actually only have one person. So yeah, no pressure. You can just have a look at them like in your own time if you want to. That might be easier. Um, does anyone have any questions? Okay. In that case, I'll just say thank you. Oh, sorry about that. Just say thank you very much for coming. Good luck for your revision. And if you could give some feedback, that'd be really good. Um, sorry, I'm just going to get to the end. Okay. Thanks, guys. Let me just put the feedback slide on. Oh my gosh. There we go. So if you could scan the QR code or go to the Google Forms link, that would be really helpful. And good luck for your upcoming exams. I hope you'll do really well. Okay, thank you.